Great. So I'm Todd Treangen. I'm an assistant professor in the computer science department, and I'm today going to share with you some results and research specific to screening of DNA that can be harmful to human hosts. And I want to thank Angela and K2I for this invitation to speak today. So just to give you an overview before I kind of jump into it over the next 12, 13, 14 minutes, is just give you an overview of what my group does, and I think this slide can summarize this reasonably well. I think a lot about software engineering, data structures and algorithms, and then infectious disease tracking and analysis. So we really do, invest heavy focus on software engineering, building tools that others can use, whether this be in the government or just other scientists that would like to use our code to analyze data. But in addition to it, to that, we focus a lot on how do we better detect and track pathogens, bacteria, viruses that can be harmful to human hosts. Sorry, going backwards. Cool. So just to give everyone a background, this is not going to, I should be able to. Shouldn't be a ton of jargon in this talk. I just want to kind of say a couple quick things. Just keep in mind. Genomic data, when I speak of it, I'm really talking about documents of evolutionary history, just all the DNA that encodes all of life, and its organism contains hereditary information. And so really I think a lot about AGCs and Ts and comparing these documents that contain AGCs and Ts and try to develop novel ways we can compare them. But in addition to this, a lot of what I've done now that we have kind of haystacks of genomic data, given the costs have dropped to actually generate the sequencing data, is trying to identify things that are hidden in this volume of data. So in this plot, what we're seeing is over since I started my PhD in well before, so the early 2000s till now, the cost to sequence the human genome has dropped from $100 million down now to under $1,000, which has really boosted the amount of available data, genomic data, which is again is our AGCs and Ts. So this is really a key point to think about as I push forward through this talk, is that the availability of data has really exploded. But importantly, it's not just the ability to read DNA that has improved. It's we can now write and edit DNA, and so we can just reading and writing. Improvements have happened over the past 20 years and beyond, and so we live in this really specific moment in time now. We have volumes of data available, and also we can generate our own data. We can say, I would like to modify this genome sequence of this organism in a way that could be beneficial or could be unintentionally harmful to someone. So really keep this in mind. We have both the read and write capabilities now for genomic information. And the writers, the editors, actually they're desktop machines that you could fit on a desk and say, please generate this set of AGCs and Ts. And so this is a really important kind of concept for the remainder of my talk. But before we get started, just to keep things informal, this is, again, I'm going to ease into everything, is a lot of what I think about when, for the SeqScreen is looking at these sequences A, B, C, and T, A, B, and C, which one of these are harmful? So I'll pause and ask for an answer and just kind of in jest, but really a lot of what the software is going to do is going to look at these three sequences and try to make a determination which one of these is harmful to a human host, right? And it could be all three, it could be none, it could be maybe the answer is there's not enough available information, but that's all the software has to consider, all of these kind of answers. And so this is a lot of, let's say, for example, the answers are that first sequence is something that's harmless. The second one is actually a sequence that's found in toxins that are harmful to human hosts. And the third one could just be there's just too short. You can't make a determination. But high level, this is what SeqScreen is doing. It's looking at AGCs and Ts of various lengths and try to determine and predict could this have any indication that this is harmful to a human host, for example. And I'll get into what I mean by harmful, but for the purposes of the talk, this could be a toxin, this could be part of the SARS-CoV-2 genome that we know, obviously we want to be able to detect. It could be a lot of different variables here that determine if something's harmful. So one thing, just kind of keep it, in addition to what I just said, context is important. So when I was trying to get to a website, I got this reCAPTCHA image, and it wasn't letting me advance. And I don't know if you can see it on the screen, but I was convinced I identified all parking meters. But in fact, they wouldn't let me advance. So you're missing one, essentially, until I clicked this mailbox, right? So for some reason, I was convinced that mailbox was a parking meter. So I clicked it, and I was able to go. But this is really important. I immediately looked at it and said, no way that's a parking meter, even if it kind of looks like one, because it would never be located in the middle of a field of grass. That doesn't make any sense. So for SeqScreen, it's going to do something similar, where it's going to use the biological context to determine, is this something that is harmful? Because there's a lot of sequences that could look on their own as maybe harmful or not, but you really need to consider the biological context. Similar to this reCAPTCHA code, if it simply would have looked at the surroundings and said, parking meters are not found in the middle of fields of grass, it wouldn't have kept asking me to select that thing. So that's a really important point here. Okay, and this is a figure from a paper I published in PLOS Pathogens, and the reason I'm bringing this up is just kind of highlighting some of the context that one could encounter inside of a pathogen, a bacteria or a virus that's harmful to us. And so essentially, what we're seeing are different sets. If you look at any individual box, it's different types of biological contexts that you can encounter inside of a pathogen. And then on the x-axis, you're going to see how difficult is to kind of identify this context. And on the y-axis is how tightly associated they are with something being harmful to a human host. So essentially on the y-axis, if you go up, known toxins that are found in certain pathogens like Clostridium botulinum and Bacillus anthracis, et cetera, are very harmful. So we know if we see them, they're going to be harmful. We need to detect them. But then there are other things in the middle here that they're kind of more in the middle ground. Well, it depends. Depends on the context. Depends on, we need to find them. But also, unfortunately, identifying all of these middle cases where there'll be combinations of certain AGCs and Ts that you see across the genome that can influence this middle ground that makes it a harder computational problem. So unfortunately, the known stuff, it's pretty easy, but that's not capturing all of it, and you can't just look at that. And of course, there are categories of things that we know that occur everywhere, and we know that those can never be harmful. So they're just different categories to think about when you're thinking about context and biological context. So what is SeqScreen exactly? So essentially, we're really thinking about a classification tool, again, that can look at a set of AGCs and Ts and output some type of helpful information to someone that would like to make a determination on whether someone should be allowed to manufacture this DNA sequence, or someone's looking in their environment and want to know, is there anything harmful in this environment there might be someone that's immunocompromised and we want to keep them safe, or there could be many other reasons for this. But essentially, given some strings of AGCs and Ts, it's going to look at and identify functions that could be concerning, and also look at and determine the context using predictive modeling that I'll get into. But before I do that, there's a concept I want to introduce called FunSoCs, and this is, so there are FunSoCs on this slide, but that's not the type of FunSoCs I want to talk about. There's actually something called functions of sequences of concern, and that's, it's kind of a mouthful, but this is through biological collaborators, biocurators that we had working on this problem, they came up with this concept to collate sets of functions that pathogens use to harm us. So that's what FunSoCs are. And we have 33 of them. I'm not going to go through all of them right now, but there's 33, and these 33 FunSoCs are essentially, you could think of them as the entirety of ways that a pathogen, a virus or bacteria can negatively affect us, okay? So that's step one. We've kind of created these categories over the last 5 years of this project to group together what are all of the ways a pathogen can harm us. And then in addition to that, we work with them, kind of human-in-a-loop training to go identify sequences that would fit each one of those 33 FunSoCs, meaning we needed to generate training data sets since nothing really existed, and we wanted to have a gold standard data set to work with this. So this is a really important point for consideration here. So we're iterating over this. We have candidate sets, and then we go to the literature. They find candidates, and they send it back to our software, and we kind of iterated over 4 or 5 years to develop training data set for each of these categories of concern so we could predict them. And just to give you some context about what was done previously to this, the prior best known database out there was something called VFDB, Virulence Factor Database. And if we look at the amount of information it contains in terms of functions of sequences of concern, we have this little dot of about 500 total proteins in a database of millions. We only have about 500 that have anything associated with functions of sequence of concern versus our curated database that we've developed that has 12,000 examples. So while that doesn't seem like a massive number, that did take a lot of effort, and it is quite a bit more comprehensive than what's out there currently. So we have this set of 12,000 sequences that we can start using to train our models on. And then just high level here, we have, again, I said 12,000, so we have 12,000 positive sets of sequences that are associated with something that would be harmful to a human host. And then we have about 80,000 negative examples, meaning 80,000 examples we can feed into our classifier to say these are known to not negatively influence a human host. And then we have a couple of different machine learning approaches, which I'll now briefly review, given the time constraints, on how we could actually predict these. So high level, model one is we're using a balanced support vector classifier plus a neural net here. And the idea is we predefine features that will characterize each of these functions of sequences of concern, so for all 33 of them, and we identify them and we feed that to our classifier, and then we let it then look at all the feature space of any sequence we would send into it. Going back to that pop quiz example, we would label those with FunSoCs and gene ontology or functional terms, and then allow it to place it into one of the 33 categories. The second example is some multitask neural networks, where we would, again, this is in collaboration, of course, with a team that I'll highlight at the end, but one name I should definitely mention is Santiago Segarra in the ECE department, who helped us develop some of these ideas. So this is a case where we'll look for proteins or sequences that contain at least one signature of being harmful, and then feed it into a neural net. So this is just a multitask learning framework, and then we have a multitask support vector classifier. And the reason we're looking at all of these is, I want to highlight something here, is that we have a very limited training data set, and we are concerned for any one of these 33 FunSoCs that we would not have enough to make a robust classification. So we wanted to look at a variety of techniques and see how that would perform. Again, this had never been done before, and it was very expensive to generate this essentially hand-curated training data set. So we then combined all three of these into an ensemble classifier and compared that to each individual classifier on this known training data set. And using that information, we evaluated on a test data set. And our test data set is essentially from subject matter experts, and they would say this sequence should be in this FunSoC, and this one should be in the other one. And so what we're seeing on the plot is essentially the analysis and the result of the individual classifiers and the ensemble classifier, and the x-axis is showing each of the 33 FunSoCs. And what the main point of the plot is showing, even though I know you can't read anything on the x-axis or the y-axis, is that it varies across each of these classifiers. And each of these classifiers you can think of specifically cornering something specific to a function of concern of a virus or pathogen that can negatively affect us, right? So we want to ideally detect all of these really well. And if we look at one of these examples, we can see that there is a, toxins, antibiotic resistance or cytotoxicity, which is indicating, or there's toxins here. So essentially all of these categories are ways, again, that a virus or bacteria can harm us, but then we'll see the classifier performance really varies over each of the categories. But what we do see, if we look at the ensemble method, which is the red line, that consistently it will give us a much better performance, given that sometimes the SVC alone or the SVC plus the neural net will have high precision in one case and low recall in the other. So the idea here is combine all of these techniques and have it there to guarantee some minimal performance to make sure we don't miss any pathogenic sequences if they are present. Okay, and then just to conclude here, as I think I'm up on time now, why is this important? Like, why do we care? Well, so as part of the manuscript that's under review and it's on now in bioRxiv, is we were handed a data set from actually a COVID patient. This is a set of sequences, a sample that was sequenced from someone that was immunocompromised. and they wanted to know if there was this patient had COVID, and running all of the existing software on it, it all returned there's no sign of the virus. Okay, we ran SeqScreen on it because again it operates on individual short DNA fragments, and it found that actually SARS-CoV-2 was present through the FunSoC modeling. But the idea is it didn't care about if SARS-CoV-2 was present or not. It actually identified the FunSoCs, the functions of sequence of concern that are specific to SARS-CoV-2, and it said 3 sequences out of 300 million sequences, only 3 it identified these are concerning, and flagged it, and flagged the sample, and it turns out what we're seeing here are the sequences that were later confirmed to be part of the SARS-CoV-2 genome, and this allows and gives you some sense of why is this useful. Well, for a novel pathogen that we have never seen before, this system would be able to look at a DNA sequence and using this FunSoC model say, hey, there's a function of sequence of concern here. You may want to take a closer look at this sample. So this just kind of end on a why is this important? And then just, this is the first paradigm that's built around this functions of sequence of concern concept for pathogen characterization. And it's also the first open source software tool for DNA screening. Nothing else exists. This is the first one that exists freely online. Anyone can download it, anyone can use it. And everything before this was closed source and you'd have to pay for it, okay? And with that, I just want to thank the team. This has been a 5-year effort. Advait, Balaji, my group, has been one of the primary developers of this software. Bryce, Kille as well. And then Signature Science has been key partners in this whole IARPA-funded project, where they actually provide much of the biocreation and biology expertise, and in addition to all the key collaborators like Santiago, Segarra here at Bryce. And with that, I will pause and take any questions you might have. Absolutely, Angela. So we recently submitted, so we're recently collaborating on this idea of how can we expand this idea out to just the identification of previously unseen pathogens in the environment. Because if I conveyed anything, and I know I flew through that, but if I conveyed anything today, it's really, I was trying to impress on, this is really one of the first approaches that it could catch something we haven't seen before that we really would care about. Because most of the existing, if not all of the existing approaches, it's what do we have in our database and compare sequences to what's in the database. And this is really about identifying sequences that you don't care what they're called, you don't care if we've seen them before, but you do care if they have a function that could negatively interact with a host. And this is at all levels. So this is kind of a systems-based approach broken down into 33 individual classifiers. So absolutely, we're looking at wastewater samples currently with her. But obviously we have aspirations broader than that too. We think this can be a really useful novel monitoring tool for environmental pathogens that we haven't seen before. Yeah, it's a great question. I didn't have time to really expand, but okay, so we spent 5 years, not me, again, that's outside, but what are all of the ways? Of course, I think there is some number that you could get to. Maybe it's 50, maybe it's 100, but it's really about identifying that. So we started with the higher priority ones, the ones that but yeah, this really is, it's a literature review coupled with, NLP methods coupled with just team discussions on and of course tapping into the vast literature on the, this type of research. But yeah, I mean, essentially 32, and yeah, you can expand out to 50. And that's, I think, one of the nice things about this software is they're individual classifiers. We're not doing it all at once, meaning you could add on 10, 5, 40, it doesn't matter. We actually have antibiotic resistance as one of our classifiers. This is a really well-studied problem. And so that's part of it, but this is the first one that considers a bunch of other categories as well. Yeah. Yeah. No, absolutely. No, I think the process behind the scenes, which I didn't get to get into, was there was a ton of back and forth saying, the biologist saying this is what the category should be, and then we're going to look at, if you look at the feature space, they're mapping all on top of each other and say these are actually not distinct. We think you should collapse them. Or conversely, we see within this FunSoC very different profiles. You should split this into 3. So absolutely.
So based on the FunSoC profile, are there specific types of pathogens that you can pre-categorize? So basically, are there a set of pathogens that have FunSoC 5, 10, and 12? Or do you basically, or is there a minimum number of FunSoCs that have to be met in order for it to be pathogenic?
Yeah, great question. So DARPA has this program called Friend or Foe, and it's about looking at an entire bacteria or virus and saying, is this dangerous or not? So we've thought about, can you pivot this into something like that? And it's kind of what you're getting at. So absolutely, we were funded for an extension on this work to look into how do you combine this information into like what you would see in like a bacterial pathogen that's common, because this is individual sequences. So in fact, you see this. You see sometimes you'll see it's always these 5 or 7. So absolutely, we're looking at that now, how you could turn this into, and what patterns you see, just what do you see, and et cetera. And of course, there's many other topics I didn't cover, but certainly the mapping of this presence absence to pathogen we've looked at, and we actually showed that you can, just using that information alone, which is in the preprint, you can outcompete a lot of the most widely cited, classification tools out there. I'm sorry? Yeah, so definitely looking into that. The only concern there is the diversity of the sequences in each class, but I definitely think for specific pathogens, you could do some type of novel capture array where that would be very different than anything that's ever been done.